Thank you very much. It's a pleasure to be here and great. Really happy for the opportunity to present our research here. I looked at the list of participants to our symposium and found that there were people with very, very diverse backgrounds. So apologies to the distinguished researchers in the first rows. I want to start this with something very basic, with a crash course in chemical risk assessment, to get us all on the same page and know what I'm talking about. Chemical risk assessment, maybe not as it should ideally be, but as it's currently done, is still based very much on this guy who about 500 years ago said the famous sentence, "The dose makes the poison," which means to characterize risk of a chemical, we need to take into account the hazard, which is the toxicity, the mechanisms of action, etc., but also the exposure, the dose that we are exposed to as humans. Without hazard, there is no risk. Without exposure, there is no risk. And we do that. We do these assessments more or less separately usually. Hazard assessment, there's mostly experimental work. We assess, as said, mechanisms of action. We can establish those response curves for a toxic effect. Exposure assessment, there's dietary exposures, estimations. There's now upcoming human biomonitoring, which is a great method where exposure is measured in urine or blood samples usually. And a bit in between what we call toxicodynamics and this exposure assessment is toxicokinetics. Toxicokinetics is part of hazard assessment, but it connects to exposure assessment. It describes everything what happens to a chemical in the human body. So a chemical is absorbed, it's distributed, it's metabolized, it's excreted, and chemicals undergo different ADME characteristics, and so this impacts from one and the same exposure dose that we get different internal exposure doses at target sites of toxicity, which of course govern then overall toxicity. And toxicokinetics also is one big reason why toxicology still depends very much on animal testing. Because yes, you can do a lot in vitro. You can establish those response curves. You can illuminate mechanisms of toxicity. It's very hard to include toxicokinetics in a purely in vitro model and human on a chip. It's a very complex system of different things that happen to a chemical in the body. But I would think it is very bad that we are relying on animal testing for several reasons. A, of course, there's ethical considerations. But B, maybe scientifically more important, animal experiments come with a high cost and with a very low speed, a low throughput. And by the immense rate at which the chemical industry invents new chemicals, if we are testing them all purely in animals, we will always lag behind with toxicity testing. There's just no way that we can be fast enough. And last but not least, humans are not just big rats. There are species differences in toxicity, both on the dynamic side, but also on the kinetic side, where, for example, chemicals are metabolized differently in different species. So, at least in Europe, there's a consensus among authorities and researchers that we should try our best to reduce, refine, and replace animal experiments, which is the famous 3 Rs of 21st century toxicology. And how would we do that? One cornerstone here is the development of the so-called new approach methodologies, NAMs, that we heard so much from in the last discussion. So basically in vitro assays that with more human relevance capture endpoints of toxicity. And yes, I agree, NAMs are in development. NAMs are still toddlers, but then anyway, rats are still rats. You know? And then we take results from there And we then need to incorporate toxicokinetics as well. We can't stop with the toxicodynamics. And so we need to perform a quantitative in vitro to in vivo extrapolation of results from those NAMs to the actual human organism. And for that, one tool has emerged as key to facilitate that, which is PBK, physiologically based kinetic modeling. And now I have finally arrived to what we are doing at the Laboratory of Toxicology here. We built those kind of computational models. It's a mathematical model that represents the human organism. It's based on human physiology, which means we have organ and organ groups with assigned organ weights, with blood flows in and out. And then we parameterize these models with data from literature, with some physicochemical data that we can predict computationally, but also with some data, especially biokinetic data, that we can measure in vitro. Parameterize the model and then run it to simulate the fate of a chemical in the human body to predict internal exposure doses at organs of interest after a given time with a given external exposure. And then, whenever possible, we evaluate the accuracy of the predictions of the model against available pharmacokinetic data in humans, or if that is not available, pharmacotoxicokinetic data from animal experiments. I'll give you an example of one thing that we did, and it's great because it's also on endocrine disruptive chemicals, so it's a link to the actual topic of our symposium. You know about bisphenol A. Bisphenol A is a suspected endocrine disruptive chemical. It's regulated in many countries and also in the EU. And the regulation is good that it's regulated. Problem is, as it was banned from some products, the industry switched to use different bisphenols as replacements. And we can find them everywhere now, but we don't have much toxicity data on them, including toxicokinetic data. So we built or expanded and refined a PBK model for bisphenol A and 6 other bisphenols. And we did not use any read across there. So we parameterized the model for each chemical individually. For example, we measured the biotransformation in the human liver in human S9 fractions, which is something that we can do easily. And then we took this data from the in vitro experiments to parameterize the model. And what the model predicts then is, if you start with one and the same oral exposure dose of these different bisphenols, you end up with very different toxicokinetic profiles predicted in blood. So bisphenol A is the black one. That's the one that is regulated and of high concern. But then also you see some other bisphenols, BPF, BPE, and what is the third one? I think it's BPAF. No, maybe not. Anyway, you get the point. You have some other bisphenols that are, by order of magnitude, more bioavailable. And we don't know much about the toxicity, I have to remind you. So that is a potential toxicological concern. And of particular interest is also this bisphenol M, which is a quite weird bisphenol. Nobody knows where it's coming from, but we know from studies that were conducted at the University of Lausanne, they find it in infant urine a lot. And it behaves differently than the other bisphenols because it's much more lipophilic, so it accumulates over time in human tissue. That's also something that our model can predict. Okay, so far, classical toxicokinetics. With classical, I mean we're looking at liver metabolism here. But in the recent decade, I would say, we came to realize that there's a second organ in humans that is very capable of biotransforming chemicals, and it is the gut microbiome. So the gut microbiome can chemically modify substances that we ingest and then change their bioavailability or their toxicity or also the efficacy of drugs. There's numerous examples of that. And so this could pose a novel mechanism of endocrine disruption where you have the gut microbiome activating a potential EDC that you ingest to something more potent. And there's actually examples for that. I can think of 3 examples. One is the mycotoxin cerealinone. Gut microbiome converts it to alpha-cerealinol. Both are xenoestrogens, but the metabolite is about 70 times more potent in activating estrogen receptors. Then there's also the case of a soy isoflavone, daidzein, which is activated to equal. And one molecule that we know very well in our laboratory is 8-prenylnaringenin, which is a metabolite of isocentrihumol. And it's not, if you think endocrine disruptive chemical, those are not the first that you would think about. They are natural. So it contained in hops, thus in beer. Why did we pick out this one as an example to conduct the study on? Well, mostly is because hops is also used to treat postmenopausal constraints. And so there was human pharmacokinetic data available that we could use to evaluate the performance of our model. So let me show you this project. Isocentrihumol is a moderate estrogen receptor agonist. At the concentrations at which it is in beer, it's no harm for human health. I'm pretty sure about that. But it can be converted to 8-PN. And 8-PN is a much more potent phytoestrogen. It has a low occurrence in beer, but we do not know to what extent it is produced by the gut microbiome. So we came up with a project. We wanted to build a microbiome-competent PBK model to quantitatively predict the systemic concentrations of 8-PN that we can find after beer consumption in humans. And then we wanted to do some basic estrogenicity testing for quantitative in vitro to in vivo extrapolation. So we started by building a standard PBK model that did not involve the gut microbiome. And we did it as usual. We made a concept of a model that included all the relevant processes that can happen to 8-PN and IXN in the human body. And then we parameterized it in the usual way, including hepatic biotransformation kinetics, including gastrointestinal permeability that we measured in a KECO2 cell line. And then we took the data from the human pharmacokinetic study that was available, gave the model the same input dose that was used in the pharmacokinetic study, and then evaluated how well it performed. And you can see it here, the dots here represent the data from the human pharmacokinetic data, and then the curves represent what our model predicts. So we are a bit off. It's not a perfect fit, but in terms of PBK modeling, absolutely acceptable. And then we went on. Now we wanted to include the gut microbiome here. And we wanted to do it in a way that allowed us for a quantitative prediction, which means we did not want to work with single bacteria or whatever, but we wanted to get as close to the actual gut microbiome as we could get. And that is, the closest thing we have is feces. So we collected human stool samples and prepared a fecal slurry from it, which can be stored. And then we pooled together, I think in that case it was 10 different fecal slurries, because also human gut microbiomes are very individual. So you need to pool to have something like an average artificial microbiome, which of course in reality does not exist, but it's a good approximation. And we incubated this pooled fecal slurry with isocentrihumol, our substrate, and measured the formation rate of 8-prenylnaringenin in the samples. And then we went back to the PBK model and implemented the gut microbiome and the biotransformation by the gut microbiome. What we found is when we measured the gut microbial formation of 8-prenylnaringenin, it followed Michaelis-Menten kinetics, which was nice to see. So this is something that we would expect from an enzymatic reaction. But B is very, very slow. So per gram feces, depending on the concentrations, we can produce up to 0.4 nanomoles of APN, which is very, very little and probably of no toxicological relevance. But then we went ahead and we tested the 10 donors, the microbiomes, individually. And we found quite the difference. So most of those microbiomes, the pink ones here, as the pooled microbiome, did not produce a lot of APN, but then 2 of those donors were outstanding producers of APN. And so we thought, okay, especially for this one, for the violet one, that has, as compared to the lower samples, a 600-fold formation rate of our chemical of interest. Maybe for that one, it is of toxicological relevance. And we put back the individual transformation rates into our PBK model to evaluate then the inter-individual differences here. And what you can see is that for this donor number 17, who was very capable in producing APN, this gut microbial biotransformation doubled the exposure to the xenoestrogen APN. And now, of course, we're going on to evaluate if this is of toxicological relevance using in vitro assays for estrogenicity testing. This is not concluded until now. Also, this is a nice strategy to further use for different EDCs of interest. And this is exactly what we are doing now. We are rolling this out to test for microbial biotransformation of chemicals as diverse as fluorinated chemicals, as pesticides. We have some mycotoxins that we are looking at now. So there's a large group of chemicals, and we perform screening. And then if we would find biotransformation products, we would try to prioritize them for toxicity testing using computational predictions, and then reperform what we did here, basically with the PBK modeling and in vitro testing. Also, a very large challenge and something that would be really cool is if we could use microbiome composition data to predict biotransformation reactions that would happen in the individuals. This would be a great step towards like personalized or population-specific risk assessment. And then also we are looking into enteropathetic recirculation, which I don't touch now because I didn't mention it before. Sorry for that. So, key takeaways here. A, the gut microbiome in some cases mediates endocrine disruption. It should be looked at as a potential mechanism of endocrine disruption. I'm not claiming that this would work for every chemicals, but I know that for some it does. B, we established microbiome competent PBK models as very nice tools to quantitatively predict internal EDC metabolite exposures, which is nice to have in the scope of all these NAMs coming up. It's really a great tool to add value to those NAMs. And then also, we see how high the inter-individual differences can be related to gut microbial metabolism, and this could be of consequence for hazard of chemicals. It could explain in some examples the differences that we observe in outcome when we're exposed to one and the same chemical at the same dose. Okay, thank you a lot. Last slide. I couldn't have done this without a great team, particularly Maya and Hélène, 2 PhD students who basically generated all the data that I showed you today. And then also shout out to our collaboration partners in Wageningen and of course our funding institutions. Thank you.